Okay. Let's come back here. Welcome, everyone. We'll get started in just a second. I get the okay that we are streaming live. I'll begin. Okay, we are good. All right, record to the cloud. Welcome, everyone, to another Water, Wetlands, and Watersheds seminar. This week, we are very pleased to have presenting Dr. Nancy Denslow. Dr. Denslow is someone I've had a great fortune to work with and to call a friend. So thank you so much, Nancy, for coming and speaking with us today. So Dr. Denslow is a professor and director of aquatic toxicology in the Department of Physiological Sciences and in the Center for Environmental and Human Toxicology in the College of Veterinary Medicine at UF. She received her PhD from UF in biochemistry and molecular biology and pioneered the use of molecular techniques for environmental toxicology, especially using in vitro assays, biomarker development, and toxicogenomic approaches. So I think we're going to hear about a bunch of that today. Dr. Denslow has over 250 peer-reviewed publications and multiple awards for her research, including the Founders Award from the Society of Environmental Toxicology in Chemistry. So I think she has both our congratulations and condolences for her upcoming position as an interim chair in her department. And so she juggles a lot and always does it with the utmost generosity and kindness as a colleague and as a mentor to students. So Nancy, thank you very much for joining us today. The floor is yours.
Thank you very much. Let me go ahead and share my screen. Okay, you've seen the right one, I hope.
It's perfect.
Okay, great. So thank you very much for inviting me to present. And so today I'm going to talk a little bit about agricultural pesticides and wastewater contaminants and the challenges they present to aquatic life. And basically, I'm going to give you a little bit of an introduction. And then following that, if it's not changing here, okay, we'll try this. I don't know why it's not changing.
See if you can move your mouse around a little bit till you see it on the screen where there it is. So now click on there when you can see it. And now the button should work. Yeah.
Okay, perfect. So I'm going to start off with an introduction, and then I'm going to give you 3 different case studies, one about organochlorine pesticides in Lake Apopka, and then talk a little bit about a project that we were involved in, which was the ICOS project, and then glyphosate in South Florida and some conclusions. So environmental contaminants are a big issue because, first of all, there are natural stressors out there, including metals and sometimes even hormones, plant hormones. But in addition to that, you have atmospheric deposition, septic tanks, groundwater, runoff from urban and agriculture, industrial discharge, sewage treatment plants, and so on. And in Florida, we're mostly worried about septic tanks and runoff from agricultural and urban areas. And often you've seen these warnings, fish consumption advisories, and that's because fish will actually pick up these contaminants. The more common ones today are the perfluorinated ones and fire retardants and so on. But still, there are legacy contaminants that will affect fish, like the organochlorine pesticides. And so nearly all substances are detectable in water. So this slide was generously provided to me by Shane Snyder, an excellent environmental chemist. Maybe some of you know him. But in any case, almost anything could be found in water at minute amounts. And so, they're not a problem for health if they really are in minute amounts because we can deal with them. It's only when they exceed certain amounts that they become a problem. So this slide was adapted by me from a book called Toxicity Testing in the 21st Century, which was a National Research Council book about how we should change ways of toxicity testing. And they were promoting more biochemical assays, cell-based assays, and that kind of thing. But the idea for this figure is that you have normal cellular responses. An organism goes through life and has a normal biological function. At some point, maybe it's exposed to some contaminant and it gets taken up into the tissues and so on. But our livers are able to take care of that. And so we can detoxify these, or we can adapt, we can compensate and get back to normal biological function. At some concentration, though, you might have permanent cell injury. And then you get adverse outcomes such as mortality or morbidity, reproductive impairment, and so on. And it's trying to know what those concentrations are. And an example of that, for example, is Tylenol. So Tylenol, everybody will take that for a headache or an inflammation. But if you take too much Tylenol, then you'll have to have a liver transplant if you want to survive, because it does a big job on your liver. Okay, so that's the whole idea of what we're trying to do, is understand what those concentrations are that do permanent injury. So a very sort of relatively new concept in toxicology is this adverse outcome pathway, where the idea is that you have a chemical that initiates a pathway that once it binds and initiates a molecular event, it's sort of like a domino effect. That means that the whole pathway will follow. And for example, then a chemical could bind to a receptor at the molecular level, and then that receptor sets off a pathway in motion, changes protein expression, maybe metabolism, might have physiological changes like tissue differentiation or egg number in fish or egg quality. Vitellogenin is the egg yolk protein, should only be in females, not in males. And then that might lead to behavior changes or reproduction changes. And at the population level, it could impact the population. And so with this idea in mind, this is an example of how this works. Fadrazole is an aromatase inhibitor. So aromatase is the enzyme that converts testosterone to estradiol. And so the pathway for hormone biosynthesis starts with cholesterol and goes eventually to testosterone, and then that's converted to estradiol. And so if fadrazole is an inhibitor, you could see in this in vitro assay that it does very well, either if the tissues are from males or from females, you could get inhibition of estrogen production. And now if a fish is exposed to fadrazole at different concentrations, you could see that the levels of estradiol in the blood go down in proportion to the dose. And the egg yolk protein, vitellogenin, absolutely depends on estradiol for being made in the fish. And then that in females, that level of vitellogenin goes down. And then you could see that at the tissue level, where the eggs now no longer have the vitellogenin. This highly colored material is vitellogenin. So these are the fadrazole treated eggs. And if they don't have egg yolk protein in them, then those egg numbers are going to not be made. So the eggs that a fish pair can make go down. And then by modeling, you can then translate that to what might happen at the population level, depending on how much vitellogenin is inhibited. And so with this idea in mind, you could see that at certain concentrations of fadrazole, it could have terrific effects at the population level. So this kind of model was developed for fadrazole, but also works for trenbolone, which is an androgen, anabolic androgen, prochlorase, that is another aromatase inhibitor, or phenaramole, which is an antagonist for the estrogen receptor. So there are a lot of different ways that the same population model can work. Okay, so systems toxicology is what we do. And basically that marries traditional toxicology where you take a toxicant and you expose a fish, and you look for phenotypic change, like changes at the tissue level, histopathology, or changes in reproduction, growth, survival, and then you can also look at how the chemical is absorbed and distributed in the fish body. And so what we do is we marry that with the omics. And what an omic is a change in global gene transcription, or gene expression, and that's what's transcriptomics, or proteomics is a result of proteins, changes in the proteins in the whole body, or metabolites in the whole body. And by analyzing these very large data sets, along with toxicology endpoints and doing computational analysis, you get this holistic view of what might be happening in the fish. And I'll give you some examples of how this works in a little bit as we go through these case studies. Okay, so our first case study is with organochlorine pesticides in Lake Apopka. So Lake Apopka is near Orlando, and this north shore of Lake Apopka was used for vegetable growing farms that used to look like this. And Wildcat Lake is a reference lake in the Ocala National Forest, and you'll see where that comes in a little bit. But they used to have they used to apply 68 pounds of DDT per acre twice a year to, control pests. And when that was outlawed, they went to chlordanes, dieldrin, methoxychlor, toxaphene. Okay. People used to work these farms, and they'd be working out there, gathering vegetables and so on. And they told us the planes would fly overhead and spray the vegetables and them and their water and their food source, everything. And then, and that was an issue for them. We met with some of these workers about 5-6 years ago. And this practice ended in the 70s and 80s. And so it's been 50, 60 years since then. And some of them, most of them that were sprayed at the time have died, but their descendants are still there. And they suffer, a lot of them, from lupus. And so we talked with them, and they were very generous, and 20 of them gave us blood samples that we tested. And we got little idea of the people that we talked to, their participant age. There were a few in their 80s and 70s and so on, and their descendants were younger. And they had worked on these farms for different numbers of years, or exposed to these chemicals for different numbers of years. And in their blood, we found a DDE that was way above the NHANES values. So NHANES is National Health and Nutrition Examination Survey that just checks the general population. And some of these had DDE in their blood at much higher levels than the average. And what was really surprising to me were there were 2 or 3 people that had DDT still in their blood, because that gets broken down to DDE, and DDE is a metabolite that is, very persistent. And so it seems like, after that much time, either they had such a huge bolus of DDT that it's still there, or they're still, being exposed to it somehow in their environment. And more than that, they reported a little bit about their health, and a few of them thought they had poor health or fair health. Not very many thought they had excellent health. And they, as I mentioned, they were really worried about systemic lupus erythematosus. And, when they go to the health department, they say, oh no, your incidence isn't any higher than anybody else in Florida. You're normal, here's a Band-Aid. But when we tested their blood samples for the antigen of these 20 people, it seemed like most of them had pretty high antigen levels. And so, it's not confirmatory that they have lupus from this antigen test, but it seems like they might. Today, Lake Apopka has been remediated, mainly by dilution. And so what they did was turned over the soil so that the levels that were high at the top were sort of mixed with the next meter or so of soil, so that now the levels per gram of sediment or soil are acceptable. We use fish as a model, largemouth bass. These fish swim in water where the contaminants are found. They are very sensitive to environmental contaminants. In Lake Apopka, the state has to put 1 million fish in every year because they're not reproducing still in the Lake Apopka. So their populations may be impacted. We still find, by the way, all of these organochlorine pesticides in the fish tissues that we get from Lake Apopka. They're top of the food chain, the predatory fish. They're game fish, which some people eat, and maybe that's how some of these people are continually getting organochlorine pesticides. And then they're a good system for human health. Here's one of my graduate students a few years back with one of the fish. And so what we did, St. John's River Water Management has done a very intensive look at this area. And some years back, I think this was 2004, where they went through and they did soil concentrations of the different contaminants. This is just showing toxaphene. And you could see that there are areas that are really hot areas. And they were very nice to let us use a couple of mesocosms that they had built out in some of the contaminated areas. And we put largemouth bass in and kept them there for 4 months. This is back in 2016. And then you could see that this is the levels of these various different contaminants before they were put in. There's still a little bit of DDE in fish that were collected from De Leon Springs, which is a clean area spring. And then after 4 months, their levels were really quite high, and of all of these contaminants. So not just DDE, but toxaphene is at a very high level as well. And so what we've been doing in our lab is looking at these, trying to reproduce what we see in the environment under laboratory conditions to be able to know exactly, what we're testing and what we're measuring. So if you go out to a control site, you can very easily distinguish the females, these are the pink ones, from the males based on the levels of estradiol and testosterone in their blood. So males have more testosterone and less estradiol. Females have more estradiol and less testosterone. They both have both, right? But at an impacted site, these amounts are really indistinguishable. And so we think that there is an issue with these hormones being affected by organochlorine pesticides. Okay, so this is one of the studies we did in the lab. We were trying to, these are the mesocosm studies, the levels, that I mentioned, and we tried to match those levels in the laboratory with para-DDE, and dieldrin. And we did studies with methoxychloroquine, toxaphene, but we weren't able to measure those levels in the lab. Okay, so we were able to show that the DDE and dieldrin had the same effect on hormones as we had seen in the wild fish. So definitely those two organochlorine pesticides do alter hormones in the fish. I'm not going through all the details of how we did this, but the results of the omics, this is looking at transcriptomics, at general messenger RNA expression. And we see genes that are upregulated are in red, Those that are downregulated are in green. Here's CYP19A1 is that aromatase that we talked about already. It was upregulated in these fish. And, ESR is estrogen receptor, AR is androgen receptor. So you can see that there are receptors in the nucleus that are altered. And all these changes predict changes in endocrine, vitellogenin, hormone biosynthesis, oocyte development, and female gonad development. This is what we expected, right? Because we knew that DDE acts as a weak estrogen or as an androgen antagonist. It has both activities and other things. What we didn't know was this. I'm not an immunologist, so I wasn't expecting all of these changes in the immune system. And these are from the wild fish from, Lake Apopka, and from our laboratory fish, we got similar results. And one of the pathways here is lupus. And I thought, oh, wow, these people are, complaining about lupus, and I don't know that fish get lupus, but they have the same pathways that humans have. And these gene changes have been mapped back to the because the human genome is totally known. And so we can map genes back to the human counterparts and use human pathway analysis to try to interpret what might be going on. And I thought, wow, this is really very interesting. Okay. So also some of the biological processes that were altered in these fish, a lot of things about lipid transport, lipoprotein metabolic process, lipid. So lipids keep coming up. So now we're doing lipidomics. Here's some more lipid binding, lipid transporter activity, cholesterol transporter activity. So doing lipid analyses, and I'm not showing you all the data, we did all of the lipids, but just showing you cholesterol. Okay, so this is looking at wildcat lake fish. This is Ocala, and this is Apopka. This is maybe 2 or 3 years ago where we collected these fish. And we found that they had DDE in their brain, in all of their tissues, a lot in the brain actually. And you see that the cholesterol seems to be down in the Apopka fish compared to the control fish. But cholesterol esters, on the other hand, are upregulated. And then that reminded me of a figure that I had seen in castrate and dual toxicology, where they're talking about, cholesterol in the cell. Cholesterol is a lipid, and it's not very free. Its solubility, it's hydrophobic. So it usually exists as cholesterol esters, and it's only converted to cholesterol as it's needed to go into the mitochondria for STAR going into the mitochondria through steroidogenic acute regulatory protein is a shuttle that brings it into the mitochondria, and it's the precursor for all the hormones, and so it's needs to be made. And so remember this figure where you couldn't distinguish the males from the females. Well, we have from other data that I'm not showing you right here, evidence that the activity of STAR is totally inhibited by these organochlorine pesticides. So that makes sense because this is being inhibited. Cholesterol levels will be low, cholesterol esters will be high. So it's just showing exactly what the impact is for this particular endpoint. Okay, so case study 2 is the ICO study that we were very fortunate to be able to participate. And in this study, we're looking at sampling locations near St. Augustine down all the way down to the marine labs, the Whitney Marine Labs, and this PCR, where these are very remote locations, not much going on around here, and we expected these to be our control sites, the wastewater treatment plant, and we thought there would be a gradient of contamination, and there are a lot of sites along here that have inputs from septic tanks, and we thought, well, maybe we would see something from that, but these were supposed to be our control sites. And so we did 3 samplings and also a pre, mid, and post hurricane. Okay, so Zaman Nouri was in my lab at the time, and we developed an LC-MS method that we could distinguish all of these different panels from a single injection. So it's a panel and that we can then look for all of these different hormones. And so we had the water samples, and we also filtered them, and we spiked them with heavy isotopes so we could quantify. We purify them through HLB columns, and then we took the filters also and analyzed those and sediments at some locations. And so, as I mentioned, we did 3 different time points: March 2019, June 2019, March 2020. And these are the water samples, these are the filters. And so interestingly, several things happened. These sites, PECR, these are these control sites over here at MOPC. I was just so astounded to see that the filters from these areas were very high in hormones. And where are these hormones coming from? I don't know. So this is up to David Kaplan to give us a model for how these are getting there, right? And from where. I know that there is a horse farm in this vicinity, and maybe that's where they're coming from. And there's new developments down here, and maybe that's where they're coming from. But, here they are. And so that was very interesting. And it was also interesting that the filters had more hormone activity than the waters did, suggesting that these hormones being hydrophobic are binding to sediments and so on. And so that might explain that. Almost, these are all on the same scale except this one. And I decided to show the full scale here. And this is really quite high. So normally in other studies, ethinyl estradiol, which is the birth control pill, it's an estrogen that's part of the birth control pill, is really high. That's almost, almost 6 nanograms per liter. It's usually found at less than 1 nanogram per liter in most locations. And so as you can see here, it gets diluted as you go downstream. And it only came up in March 2020. It wasn't there in these other, these are just grab samples one day, that we go out. So it's not integrated over time. And I thought, wow, that's really high. Okay. So this was the sediments. And again, sediments from March 2019 were a lot higher. There's a lot in these PCR locations. These are control sites, supposedly. And for some reason, March 2020, we didn't see much. And I still don't understand why it seemed to disappear there. Except, again, this is a shot time point. A storm event that we did, pre-storm and post-storm, and then really post-storm. This is exactly just as the storm ended, and this is a few days later. You could see the changes in these hormones over this time, looking at both the water and the filters. Okay, so why do we care about hormones in the water? So the birth control pill, we know here we found a value of 6 nanograms per liter of EE2. We know that 5 nanograms per liter EE2 has enormous effects in fish. And I'll show you a little bit of data. And 100 nanograms per liter levonorgestrel. Levonorgestrel is a progestin part of the birth control pill, also has profound effects. For example, we did 30-minute exposures in fish of these two chemicals, and we found protein changes, proteins that have been phosphorylated that map to neurogenesis pathways and other things. And so that happens. At 5 nanograms per liter EE2, you get induction of vitellogenin in males. This was a 48-hour exposure. And you see that control males don't have much vitellogenin, but if you expose them to 5 nanograms per liter for 48 hours, you get quite a bit of induction of this protein in the blood of these fish. And what was very interesting was that this was a mixture with an anti-estrogen. It's supposed to be a very pure anti-estrogen, and didn't seem to budge, as suggesting that maybe it was coming through estrogen receptors that were membrane receptors that antagonist doesn't affect. But in any case, we are looking at that. And then you could see that in females, 5 nanograms per liter EE2 affects egg production. So these are paired males and females for all of the different ones. This is work done by Santos et al., showing that they're producing eggs at about the same rate. Oops, sorry. But then when you feed them, when you expose them to 5 nanograms per liter EE2, then those that were exposed stop, or reduce the number of eggs that they make. So it's an issue. So what we're doing, we're collaborating with Boone Prentice in chemistry, where he does in situ for looking at lipids by MALDI. So this is a tissue slice looking at MALDI changes, lipid changes by MALDI. Oh, sorry. Sorry. I get too excited here. And so we also cut out these different parts of the brain. So this is a fish brain. And this is the front of the fish. The nose would be over here. And then these are the different lobes. Yeah, fish have brain lobes just like humans. And so we had cut out individually these different parts and have done lipidomics. I'm just showing you one little slide here. And also then Gina G in Boone's lab is doing slices across these livers and making these really thin slices and putting them in the MALDI and looking at changes in lipids. And what I think is just fascinating is that, first of all, the lipid composition of different parts of the brain are different. And what that means, I don't know. And that exposure to estrogen or to a progestin, I'm not showing you the progestin work here, changes that lipid expression. And what that means, I don't know. Okay, except that these are bioactive lipids and they're made when they're needed for a specific purpose. So, and then we got similar effects when we did this. Oh gosh, okay, come on. Getting too excited here. We got similar effects when we did this, by cutting out these individual things and doing the whole lipidomics profiling. And I'm just showing you a couple of slides. And what that means, I don't know, except that these are bioactive lipids and 5 nanograms per liter E2 changes them in 24 hours. So we're trying to figure out what that means. Okay, going to the next case study, and this is glyphosate. So glyphosate is an active ingredient in Roundup, and it's mainly used for agricultural purposes. And this is just a USGS map showing how it continuously gets increased over time. But notice in Florida, it's always dark brown. It's because it's not used only for GMOs. That's what everybody, glyphosate's GMO. Well, yeah, but it's used for agriculture in general. And in Florida, it turns out that for the sugarcane industry, that if you add glyphosate to those fields a couple of weeks before harvest, that you get a better yield of sugar from the sugarcane. About a 15 to 20% increase in yield. So it's sprayed on the sugar cane right before harvest. It's also sprayed directly into streams to take care of invasive plants because there's not much else that can take care of those. Okay. So what we're going to, we have two studies, one with largemouth bass and one with manatees. So my graduate student, Maite de Maria, just graduated, so she's Dr. de Maria now, did this exposure with glyphosate and rodeo. Rodeo is the commercial formulation that is actually applied into the water for invasive plants. And so we had largemouth bass for 21 days to try to get a better idea. And so here you see that so the concentrations that we used, we targeted 0.5 milligrams per liter and 10 milligrams per liter. And this is the level that EPA says is okay to have in drinking water. Okay, so we went below that level and slightly above that level. And these are the actual concentrations. So we sort of, you know, we meant to be at 0.5, but we are a little bit below that. Okay, so that's what we actually did. All right, so this is a gene expression results, and again, trying to just show the max, you know, the top analysis without going into all the details. You can see there's quite a bit of difference between controls and the glyphosate treated in terms of gene expression. So red means that it's upregulated more than it was in the control fish. And yellow is a little bit more than the control fish. Blue means it's downregulated. So you can see these values here for glyphosate and for rodeo. We did trunk kidney in the fish, and they also have a head kidney. So the head kidney and the trunk kidney are connected in the fish. And the head kidney is more like an immune organ in the fish. And the trunk kidney is similar to our kidney. And you can see that those are not identical in the trunk kidney and the head kidney because they do have different functions in the fish, but their expression values, there are a bunch of genes that have been altered because of these exposures after 21 days, which is chronic. Okay, so we had a number of different pathways that were altered. Some immune pathways, which was what we were part of our hypothesis, that there would be immune dysfunction. We found it. How kidneys function also was affected. And then steroid hormone biosynthesis was another one that was affected. And that's the one I'm going to focus on for this talk because I don't want to run over my time. And what we found is that one of the genes right here pointed that was downregulated, blue means downregulated, is this gene that converts testosterone to 11-ketotestosterone, which is the main male hormone in fish. And so it was downregulated. But there were a lot of other genes that were downregulated as well in this hormone biosynthetic pathway. And glyphosate has been already predicted to affect STAR. Remember, STAR is the one that changes cholesterol and starts the whole thing for hormone biosynthesis. And we were able to measure the hormones directly in the blood of these fish. And in fact, we found that for 11-ketotestosterone, there was a general downregulation in all of the concentrations that we tested. And you can imagine that the bigger the fish, the more testosterone they might have. So the more testosterone, the more 11-ketotestosterone they have. But regardless of that, 11-ketotestosterone dropped. And then also estrone dropped in these fish because testosterone is a precursor for both 11-ketotestosterone and estrone, which is E1. And both hormones were affected by glyphosate. So again, you know, this is something that could be problematic for largemouth bass. All right, and we haven't published the largemouth bass piece yet, but we're hoping to soon. So she's also her primary interest was with manatees. So manatees are marine mammals, and they're most closely related to the elephant. And if you haven't been to Crystal River to swim with the manatees, it's a must. You really should go. It's really a fun kind of thing to do. And they're herbivores. And they range all the way from Brazil to the South USA. And so her interest was looking at this agricultural area near Lake Okeechobee. So all this dark green area is where the sugar cane plantations are located. And so she went down there actually and put POCEs. POCEs are passive samplers, because we didn't expect to really find it. We thought, okay, well, let's put the passive sampler there for a month. And if there's a spike, we'll see it, we'll integrate it, we'll find it, because it's supposed to break down very readily in water. It goes as glyphosate is the amino acid glycine with an extra phosphate group on it. And it's easy, for bacteria to degrade this. It goes to AMPA when it's degraded, and then goes to carbon dioxide and ammonia. So it's not a problem. And it affects a particular pathway in plants that doesn't exist in humans to make certain amino acids, essential amino acids that we get from our diet. So we don't have that pathway, but plants make those amino acids. So they do have the pathway. And so it kills the plants, but not people. So it's supposed to be the safest herbicide around. Okay, so we also took grab samples whenever we went down. We took a grab sample of water when we went to put the sampler in, and took another grab sample when we brought it home. And we found glyphosate in all of our samples every time. Okay, so Crystal River is our control site. And then this is the agricultural area. And so these are the relative amounts of glyphosate. We found glyphosate even in Crystal River, but at much lower levels. And then we did 3 seasons. We did August, December, and then March, we wanted to do March, ended up doing May because of COVID. But anyway, so it was pre-harvest, during the harvest, and after the harvest. And you could see in this more agricultural area that it seems to match the harvest. At this location it was higher pre-harvest. So this is probably indicating its use for other things in Florida and not just for sugarcane. As I said, it was also sprayed directly into these locations for invasive plant control. And so that's what we found, and AMPA is these very small amounts. So it looks like it's not degrading very well. So then we went and we looked in the literature and we found, well, if there's a lot of phosphate around, that inhibits the degradation of glyphosate, because the bacteria would rather get the phosphate directly, and the phosphate amounts down there are pretty high. So maybe that's why it's not degrading. Or if it's degrading, the input is constant, and it's degrading, and so, but it's constantly there. Okay, we always found it at every sampling. And more interestingly, it was also found in the manatee blood. So she sampled 106 manatees from all locations, but manatees swim, and so you don't know where they pick it up. But 106 manatees were sampled, and 56% or 56% of them had glyphosate. And the plasma samples she had were from a number of different years. And so you could see that the trend, this is a log scale, the trend is to go up. And even in Crystal River, which is the blue line, the trend is going up over the years. And that makes sense because we know that the use of glyphosate in agriculture has increased tremendously since 1994. It was a 15-fold increase in use between 1994 and 2014, data we were able to find. And then there's this group here in orange that appears to go down over those same last 3 years. We only had 3 years of those. And these were from Georgia, but these manatees tend to be more in the St. Johns River. And the application of glyphosate for aquatic vegetation was reduced tremendously over those last years. And in fact, the last year that we had the manatee samples, Fish and Wildlife didn't even apply glyphosate at all. So it looks like maybe there's a trend here towards decreased amounts when glyphosate is not in the environment, but that's something that needs to be continuously monitored. We don't know if that's a trend or not, really. Okay, and then she also did sampling with the manatees, where she was able to get the white blood cells. I mean, so you can't take a manatee and put it in a tank and do an exposure to a manatee. It's not a possible thing, but you could get the blood samples, and from the blood cells, you can get the white blood cells, and then you could culture these. And so she did that. And one of the things you could do with white blood cells is look at how well they respond to a challenge for a mitogen. And the blood cells are supposed to, divide quickly to take care of that mitogen. And what she found was, and so that's a good kind of reaction. And what she found was that with increasing glyphosate applied to the in vitro cell assay, that this ability to attack an invader disappeared. And so that we think is immune suppression. So, it was only at the high dose, but these were, placed. So we don't see those kinds of amounts in the manatees, but we see chronic levels. And so the difference between an acute level and a chronic level, I don't know if it's really contributing to immune suppression in manatees, but it's a suggestion that it could be. And so with that, I would like to conclude that endocrine disruptors are present in the environment and likely present in aquatic wildlife, especially with chronic exposures. And that these, for example, the organochlorine pesticides are persistent and can still be found in the north shore of Lake Apopka and in the lake itself and in the biota. And I think it can cause hormone biosynthesis disruption. And maybe that could explain why every year they have to put 1 million fish into the lake. And high volume pesticides like glyphosate are present in the environment despite being very polar. So they're exactly the opposite. They're extremely polar. And so, and chronic exposures could cause immunosuppression. And with that, I would like to thank the people in my lab that have contributed to this work. A bunch of people have already left, and postdocs and graduate students, and a bunch of collaborators. And I wish I could have, had more room to name everybody who was involved, and happy to take any questions.
Thanks, Nancy. Let's give Dr. Denslow a round of applause, either real or Zoom claps. And thank you so much. Move this out of the way. Great, thanks, Nancy. So we'll start with any questions from those in the Zoom room. I'm going to pop over to YouTube and see if there's anything over there. So if you have a question, please unmute. Christine, I see. Go ahead.
I don't have one quite at the tip of my tongue. I really enjoyed that, Nancy. I guess maybe I will ask one, which is, how do we bridge from kind of these individual like lab scale kind of analyses to understanding like population level consequences? Like, what do you see in the case of, say, the manatees or the fish in Lake Apopka? Like, how do we get to like the population scale dynamics? What is that step that you see that's needed to kind of to link from your mechanistic work to those? Yeah.
This is where we need ecologists and modelers involved, because, some of these organisms have long lives. And so you can't really quite do the experiment. Although with ethinyl estradiol, the whole lake experiment was done in Canada, where they have these lakes, and they actually put 5 nanograms per liter of ethinyl estradiol into a lake and looked to see what happened to the populations. And it was very interesting. So depending on the fish, like fathead minnows had a big issue. But other, fish populations didn't really, they were able to survive. And then when they quit using it, then the fish populations came back. So it looks like it might be temporary issues, which is a good news, because that means we can clean things up and, and get back to normal. And so, yeah, but this is where the population, modelers are extremely important, because it's only through their models that we can know.
Yeah. Okay, thanks.
Thanks, Nancy. So I'm thinking about this explanation you gave of glyphosate or Roundup or rodeo, and it's like, oh, it breaks down to AMPA, and then it breaks down to CO2 and ammonia, and it's so widely used from everything from lawns to large-scale agricultural to ecosystem management. And then you said that there's this pathway, it's only in plants. And
Right.
definitely, like, I'm naive on all the like biochemistry. And then you said it actually changes all this stuff in a marine mammal and a fish. So can you just help us, like, for there are students on here who are maybe not so aware or understanding all those metabolic pathways. Can you just kind of, well, for me, can you dumb it down for me? How is it that it is sort of explained as sort of one kingdom, plants, and it's having all these other effects?
Well, I think these are non-target effects. And so while that particular pathway, there's certain amino acids that we must get from our diet that we cannot synthesize normally. And so these are through the shikimic acid pathway in plants. So plants make these amino acids. They're tryptophan, phenylalanine, tyrosine. I think there are 3 of them. And so we have to get those from our diet. So that pathway doesn't exist. And so maybe at very low concentrations, it affects the plant, but it doesn't affect us, right? But these are non-target, and so.
So it's just sort of like the law of unintended consequences. It was targeted to disrupt a particular pathway that doesn't exist in animals, and it doesn't. It just has other non-target effects that were just not considered. Yeah.
And so, the short-term assays, acute, didn't show an effect. And you could see that, we need, pretty high concentrations to see the effects that we see. We don't see those effects at very low concentrations. We see them only at high concentrations. But then the issue is, what about chronic exposure? So I don't think that a single exposure is going to do you any harm at all. Right? But if you're exposed to this chronically, day in, day out, if you're a manatee and you swim in the waters, you can't quite get away from it, or a fish, then maybe that does have unintended consequences. And I would suggest, we haven't done any, glyphosate has come up as carcinogen. And I'm not suggesting that, because I don't have any data that would support that. But some people have, been awarded funds, monies, because they, were able to show that daily exposure to this caused their cancers. But I don't know that it does. And my data doesn't show cancer at all. It shows immune suppression.
Right.
But that could be related to cancer, because the immune system is always going around making, taking care of that. So.
Right, exactly. Kathy, please go ahead.
I seem to remember many years ago that one of my colleagues in zoology did a study showing the effects of increasing nest temperature on sex determination, I think in crocodilians. And so I found myself thinking about effects of temperature in these situations where perhaps temperature might increase the rate at which a pesticide deteriorates, or it might increase the impact on the pathways, or there may be conjunctive effects across 2 different kinds of stresses like that. So I guess the main question is, how is temperature, especially considering climate change, likely to affect the kinds of results you've seen?
That's a really interesting question. We haven't done any experiments with glyphosate or any of these with temperature. We've done some temperature with other contaminants that hit mitochondria and have found interactions. And so there could be, I don't know. We haven't looked at it, but very, very, very interesting question. Yeah. Should be done. Could be.
Well, one good thing, Nancy, for you and all your students and postdocs is that if when glyphosate is phased out, then the next thing will come, and then the next, so there's always another organic
I know.
chemical on the horizon, and
Yeah.
particularly as plants begin to develop, like adaptations around some of those pathways of plant control. So I expect we'll be hearing more and more about this in the future. So everyone, we've passed our time, so please give another round of applause to Dr. Denslow. Thank you so much for joining us today. Really great to see you. And everyone else, we will see you next week. We have a change in schedule. We had Dr. Bob Weisberg from USF was unfortunately had to cancel, but we're going to get Dr. Sherry Larkin, who is the director of Florida Sea Grant. Is that right, Christine, right? So we're going to hear a really awesome talk about what Sea Grant is doing around the whole state. Thanks, everyone. Be well. Take care of each other.